Welcome back to my channel. In today's video, I'll be talking about that how we can visualize a bacteria with the help of microscope. So the first one, which is very much important, that is staining. And the most important staining is gram staining of bacteria. Now, in case of gram staining, there are few steps which we have to follow. The first one is apply crystal violet on the bacterial smear on a glass slide. This is the first step. Then we have to add iodine solution, also known as Gram's iodine solution, onto this so that it helps to retain the stain in the bacterial cell wall. Remember this bacterial cell wall. Why? Because this will form in step 3 a crystal violet iodine complex, or in short, I should write it like that, crystal violet iodine complex. Now, in the third step, the decolorization step. Why? Because we will use a decolorization agent or decolorizing agent known as ethanol, and that is 95 to 99% ethanol should be used to decolorize the bacterial cell wall, because it is retained inside the bacterial cell wall. And then the counter stain called safranin should be added after this decolorization step, and finally we should wash it with the help of distilled water. And air dry the slide, and finally we can watch it under the microscope, the light microscope. Now, one important thing is the third step, when we use the 99% ethanol to decolorize the primary stain, which is actually complexed with the Gram's iodine. In this case of gram positive bacteria, when the ethanol is used, it basically shrinks the pores of the highly cross-linked PDG layer. Okay, so ethanol generally shrink the pores of highly cross-linked PDG layer, and due to that the crystal violet iodine complex will be retained in the cell wall. It cannot be washed away with the help of this ethanol, so it is retained in the cell wall. And when the primary stain is retained in the cell wall along with the iodine, it will form a much more stable complex. When we will use the counter stain safranin, the cell will not take up the counter stain, and we will see the bacteria as a purple color bacteria under the microscope. So that is the indication of gram positive bacteria. Remember one thing, when we apply this gram staining and the cell remain purple or visualized as a purple cell under the microscope, it is known as gram positive bacteria. Okay. Now, in case of gram negative bacteria, it looks like a pink or light reddish color under the microscope. Why? Because during the decolorization step, the ethanol readily removes the lipids from the lipid bilayer, which is present in the outer membrane of the gram-negative bacteria. So the lipid from the outer membrane are washed off. So there is no barrier right now. And then the PDG layer in case of gram-negative bacteria is very thin as compared to the gram-positive bacteria. So due to the presence of this thin PDG layer and without the lipids from the outer membrane, ethanol will expand the PDG layer much more, and this will helps to wash away or remove all the crystal violet iodine complex. Okay, so when we will apply the counter stain safranin, there is no crystal violet iodine complex now present in the cell wall. So only the safranin can stain the cell, and which will look like pink under the microscope and indicates the gram-negative nature of the bacteria. So this is basically the basics of the gram staining to differentiate between the gram-positive and negative bacteria. Thank you so much for watching this video.